Hello, everyone, and welcome. I think we're about to get started here. Just wait another 30 seconds or so. In the meantime, just a few housekeeping reminders. This webinar is live and recorded, as mentioned previously, excuse me. Please remain muted with cameras off for the duration of the webinar, and after the presentation, there will be a brief Q&A session, time permitting, for any questions which should be submitted in the chat window in the bottom right of your screen. If we don't have time to go through all questions, we'll do our best to at least follow up after the webinar concludes, and the recording of this webinar will be available and accessible via Microfluidics website as well as our related channel partners. So again, welcome to all those who have joined us this morning. We appreciate your company. Thank you very much for attending today's webinar titled The Comparison of Biotest HSA and Recombinant HSA for the Preparation of Nano Albumin Bound Paclitaxel using Microfluidizer technology. So again, welcome. My name is Matt LaFerrier, and I'm an applications engineer at Microfluidics, applying microfluidizer technology and knowledge to a wide range of applications by working closely with customers in numerous proof of concept, process optimization, and scale-up tests. Myself aside, we have two presenters for you today. Our first speaker is Liye Maeyama, Dr. Liye Maeyama from Biotest AG. She is Biotest AG's global product manager and is currently focused on the assessment of various characteristics of albumin and its effect on albumin excipient applications in nanoencapsulation and induced pluripotent stem cells. Our second speaker for you today is Dr. Cologne University. Since August 2021, Carolyn has been a senior scientist at the Research Center of Pharmaceutical Engineering, focusing on manufacturing and characterization of nanodrug delivery systems. For today's webinar, we have a brief agenda for you, so we'll go through who Microfluidics is as a company and how our technology works and relates to this application, which will be followed by Liye's discussion of Biotest HSA product and their quality data, before concluding with Carolyn, who will dive into her DOE studies and quality results from RCPE before the presentation concludes with the previously mentioned Q&A session. So, who is Microfluidics? Microfluidics has produced high shear fluid processors using our proprietary fixed geometry interaction chamber technology since being founded in 1982. Headquartered outside of Boston, Mass., microfluidizer processors are designed and used for research and development projects, as well as manufacturing challenges in an array of applications, but specifically active pharmaceutical ingredients, vaccines, and coatings, to name a few. In 2011, Microfluidics was acquired by IDEX Corporation to create our MPT platform, where we continue to develop extensive machine design and application experiences. Our international team will help answer any questions or challenges that you may have, regardless of where you are in the world. Here, you can see that we have some other applications aside from the one being focused on today that microfluidizer technology is not only commonly utilized for, but where it can also deliver industry-leading results by producing small, uniform droplet and solid particle sizes and distributions. Aside from today's application, cell disruption is another great example where microfluidizer processors excel at producing desired product with high rates of protein recovery. So how do we achieve such fantastic results, you may ask? Microfluidizer processors operate by loading material into the inlet reservoir, which you can see here. Excuse me for 1 second. So the inlet reservoir is here, where your material will be loaded before being pulled via this constant pressure intensifier pump into our fixed geometry interaction chamber at pressures of up to 30,000 psi. At such pressures, the material experiences incredibly high shear rates and impact forces that are consistent and uniform, Now, by combining such high pressures with a fixed geometry interaction chamber design, this ensures that all of the material is processed under the same conditions. Downstream from the interaction chamber would be a heat exchanger or a cooling coil to control product temperature from processing. And this is utilized before collecting right at the product outlet. Now, for this fixed geometry interaction chamber, we have two chamber designs. We have the the Y-type interaction chamber, which utilizes a unique path where the material splits into two streams, each of which is is forced through a microchannel before impinging on each other at this center point. The combination of microchannels, pressures, and impingement provides intense micromixing before the product exits at this low pressure outlet. Unlike the Y-type chamber, the Z-type chamber at the bottom of the slide only utilizes one microchannel instead of two, but it still experiences incredible shear forces and impact forces after the material impacts against this back wall before exiting in a low pressure outlet. The Z-type interaction chamber is utilized in today's application and in other applications that involve solids because this design is less prone to clogging while processing. Now, one guaranteed exceptional quality that Microfluidics Microfluidics can apply for their customers is the guaranteed scalability factor of our processors. On the left side, we have our laboratory or pilot scale product line interaction chamber designs that we previously discussed. And on the right side, we have our production scale interaction chamber designs. To ensure scalability, these microchannels from the lab scale are essentially placed in parallel, as seen on the right, allowing for increased throughput of material. Utilizing the same interaction chamber geometry and microchannel dimensions, these interaction chambers and our overall technology should deliver consistent scaled results regardless of material quantity or machine size. Here, we just have a brief view of our beautiful line of machines. We offer an impressive range of microfluidizer processors capable of processing as little as 1 milliliter of material using our LV1 lab scale microfluidizer processor to processing potentially thousands of liters using our production scale microfluidizer processor. 4 out of all units seen on screen here are cGMP capable as well. Please, that would include the bottom 3 units as well as the top right unit. Please reach out to Microfluidics team to further inquire about our products or about our company or anything that you think we might be of help answering, or place your questions in the chat window below to review at the end of the webinar. Thank you for listening to my portion of this presentation. And now I will give the reins to Liye and hers. Thank you.
So thank Matt. Thank you for joining. It's great that you are all here. And my name is Liye Maeyama from Biotest, and I would like to welcome you to the session Biotest Human Serum Albumin, a multifunctional excipient. So I work for Biotest AG, and we are specialized in the development and production of vital medicine purified from human plasma. Biotest AG is a global company based in Germany, near Frankfurt, founded in 1946. And we are specialized in treating disorders in the immune system, blood forming system, and intensive care. And our company is covering the whole range from plasma sourcing, preclinical, clinical development, and manufacturing. And of course, we also do the promotion, the marketing, worldwide marketing and distribution. And today, I would like to talk about 3 main topics. First, starting with the albumin molecule and to explain the multifunctionality of the albumin molecule. And then I would like to give you the summary of our product, Biotest HSA. And I would like to show you the data we collected on quality to support the high purity of Biotest HSA. The albumin molecule and its multifunctionality. So albumin is a molecule which is performing complex functions. It is the most abundant protein. More than 50% in plasma is albumin. It is non-immunogenic, highly biocompatible, biodegradable, and very safe. The albumin chemical structure, it allows binding with many different drugs. And the serum half-life is between 12 and 20 days. We say in average it's 90 days. And this feature means it protects potential drugs from elimination in vivo. And of course, it's also improving the pharmacokinetic properties. And albumin has also the characteristic of interacting with receptors, which are overexpressed in cancer cells, active, and this makes of course, an active targeting of the disease possible without ligands. And you can see here the albumin molecule. It is a heart-shaped molecule. Just a moment. Cannot see the laser pointer, sorry. But you can see here that it is composed of three main domains. And especially important is the cysteine 34, which is very important for the antioxidant property. Albumin has oncotic and non-oncotic function. I think the known property is mainly the oncotic characteristic. So albumin is maintaining the intravascular colloid osmotic pressure. But it has many non-oncotic properties, which is solubilization and the transport of ligands. It is the main circulating antioxidant. It has anti-inflammatory characteristics and immunomodulation characteristics. It is contributing to the maintenance of vascular integrity, to the acid-base balance. It has antithrombotic, anticoagulant properties. It serves as a protein reserve. It supports wound repair by metabolic and functional activities. It has detoxifying properties. For example, it can reduce cytopathic effects of bacterial toxins. And it has also, it is able to reduce the photodegradation rate of folic acid. And you can see by all these functions that albumin is actually an amazing molecule with many different important functions, physiological functions. Now I would like to explain you the product Biotest HSA and the main difference or the common features with the pharmaceutic. So we, from Biotest, we offer two different product lines. One is for the therapy, the green one. We have albumin, which is the therapeutic, so the medicine. And it goes directly to the patients. So we have the drug license in more than 50 countries, and this product is used for more than 30 years. We have another product line, which is the blue one, which is Biotest HSA, and this is for manufacturing use. This product is being used for manufacturing, producing different kinds of products, which eventually goes back to the patient. And because it has a pharmaceutical grade, it is safe for the patient. So Biotest HSA is a multifunctional excipient for sophisticated applications. It serves as a stabilizer for drug and vaccine formulation and cell therapy. It is a drug carrier for targeted delivery. And our topic is today nanoencapsulation, so nab-paclitaxel. It is also being used for medical imaging. A major application is also cell media. So HSA is used as an ancillary agent for cell media to cultivate cell and tissue, stem cells. It is being used for cryopreservation media and also for media for in vitro fertilization. One very exciting area of research is, as you might know, is the creation of artificial organs. And back in 2019, Israeli scientists, they were the first group to create the 3D printed heart using human cells. And how they did is they took induced pluripotent stem cells, cultivated them in a cell culture containing human serum albumin, and these induced pluripotent stem cells are differentiated to cardiomyocytes. And with these cardiomyocytes, they did the 3D print of the heart. So Biotest HSA is a product. We have two different packaging sizes, 100 milliliter, 20%, 5%, 250 milliliter. It is, as I said, for manufacturing research use only. It is produced in a GMP environment. So we have pharmaceutical grade. We comply with the European Pharmacopoeia monograph. We have all the same manufacturing documentation as the medicine albumin, and the product is batch released by Paul-Ehrlich Institute Germany. And this is the authority in Germany which is also in charge for releasing the vaccines. For example, the COVID-19 vaccine from BioNTech Pfizer is also being released by Paul-Ehrlich Institute. We are free of leachables, nitrosamines, latex polyvinyl chloride, also free of titanium dioxide and melamine, and BSE, TSE relevant materials. So we are providing our clients an overall service. So we have a global regulatory network, and we can also provide you with all the documents which you need for the finished good to submit for drug license. Now I would like to give you a summary on the quality data we have been collecting. And these quality data would support the high purity of Biotest HSA. So we have a lot of cooperation with scientific and research institute. So what we did here is with the University of Frankfurt, we did an analysis of residual small molecules in various albumin preparations. And you can see in the graph the intensity ratio and number of additional signals. The lower the values are, the higher is the purity. And in comparison to ABC, we can see that Biotest HSA tend to have a high purity. These are the same data which are measured in NMR. So you can see the visualization by diagrams, and one signal would represent a small molecule. And comparing Biotest and other albumin preparation, you can see also here the high purity of Biotest HSA. What we did internally at Biotest, we checked some of the specification parameters. And what we found is that the parameter aggregates, dimer polymers, aluminum, ethanol fragments, and sodium, we can also see lower values of these parameters and conclude that we have a higher purity in comparison to the other albumin preparations. Another research cooperation we had is with the Fraunhofer Institute, research organization in Germany. So we made an analysis of fatty acids. Fatty acids are playing a role for stem cell cultivation. And we could find that Biotest HSA has a lower concentration of fatty acids, especially for palmitate, palmit oleate, and Cysteine oleic acid. We found significantly lower concentrations. So another parameter we investigated, this was done in cooperation with the University of Graz. We checked the stability in terms of the oxidation of cysteine 34. Cysteine 34 is an amino acid in the albumin molecule, which has a free thiol residue, and it has 3 different stages. One is the reduced state, redox state, which is represented here by HMA. Then we have the state of reversibly oxidized cysteine 34, which is here, you can see in the graph of HNA1. And the third graph, HNA2, this is the irreversibly oxidized state of cysteine 34. And we can see that by statistically, for ABC, there is a trend that the reduced state of cysteine 34 is decreasing with the time passing. And in case of Biotest, even after expiration of the sample, after 1,200 days, 3 years, it is relatively stable. So meaning the cysteine 34, it's not so much oxidized like ABC. We have to say there is a lot of variance with Biotest HSA, and we need to do more investigations. So as a summary, we have high purity in terms of the 5 lows. So we have a lower content of aluminum, ethanol, aggregates, fragments, and sodium compared to other albumins. We have lower concentration of fatty acids, especially palmitate, palmit oleate, and Cysteine oleic acid. We have a lower number of small molecules, which was done in the nuclear magnetic resonance spectroscopy, and we tend to be more stable against oxidation. So I would like to express my special thanks to the RCPE team, especially to Caroline Tetitskaya for the great experimental work on the nanoencapsulation nab-paclitaxel formulation, Ramona Jatler for advising throughout this project, and Eva Faulhammer. And also my special thanks to the Microfluidics team who made this platform available for the webinar, and especially Casey Silva, who did a great job in coordinating the joint webinar, Christina Schneider, Matthew Baumer, Steve Messer, Sue Young, Matt Lafier, to make this webinar possible. Thank you very much for your attention. And now I would like to pass to Caroline to present the work she has done on the nab-paclitaxel using the microfluidizer. So, Caroline.
Thanks, Liye, for introducing myself. Hello, everyone, also from my side. My name is Caroline Tetitska. I am Senior Scientist at Research Center Pharmaceutical Engineering, or RCPE, which is located in Graz. Within this presentation, I will first of all present you our research center and then talk about our study, which was performed together with Biotest and Microfluidics. So we compared here the use of Biotest HSA with a recombinant HSA for the preparation of nab-paclitaxel. So what is RCPE and where are we located? RCPE is a research center located in Graz in Styria. This is in the south of Austria. And we are global leaders in pharmaceutical process engineering. And by optimizing our products and processes, we help our partners to develop and manufacture innovative medicines. Our science enables tomorrow's medical discoveries and improves patients' lives worldwide. We are currently more than 160 employees of 24 nationalities and have more than 150 partners, industrial as well as scientific partners. We so far published around 600 papers, have a turnover rate of around 14 million euros, and are equipped with devices of 19 million euros. Our main research fields include resilient supply chains, digital tools for complex products, manufacturing, advanced formulations, as well as better regulatory approval models. We are composed of 3 major areas. Thereby, Area 1, or Science of Quality, deals with digital twins, modeling and prediction, process control and PAT, advanced quality assurance, analytical and artificial intelligence, as well as quality by design. Area 2, or Advanced Formulations, focuses on oral dosage forms as well as new modalities, excipient engineering, lipid-based systems, personalized medicine, stability of dosage forms, biopharmaceutics, and material science. Area 3, or Next Generation Manufacturing, where I am part of, focuses on high-speed manufacturing, nano-manufacturing platforms, advanced processing technologies, novel sensors, personalized and on-demand manufacturing, as well as greener technology and sustainability. With our infrastructure at RCPE, we can resemble real-world production settings and can provide practical solutions to your needs. We have at the moment state-of-the-art facility of around 800 square meters, a dedicated pilot plant, and with pharma standard clean rooms. There OEB class 3 to 4 substances can be used, but also highly potent substances like paclitaxel, for example. We have a dedicated vaccine room and an extensive selection of different equipment at different scales, and also a modern analytical laboratory. Now coming to our study, currently around 65% of cancer drugs are of natural origin. Thereby, paclitaxel is one of the most important drugs against cancer. The anti-tumor action of paclitaxel was thereby first described in 1967. However, until an approval of a formulation, it took around 25 years. In 1992, then, the FDA approved Taxol, which is a non-aqueous solution of paclitaxel dissolved in cremophore L and ethanol. As can be seen on the right image, the sale of Taxol increased after approval, and by 2000, Taxol was the best-selling cancer drug of all time, with annual sales of $1.6 billion. However, the treatment with Taxol shows many severe side effects. These include hypersensitivity reactions and also neutropenia, as well as sometimes the treatment of this formulation leads to the death of the patients. Hence, there was a need to develop another formulation with paclitaxel, which shows increased safety and could also encapsulate paclitaxel as it is poorly soluble in water. In 2005 and 2008, FDA and EMA then approved the first paclitaxel nano formulation, the so-called nab-paclitaxel or Abraxane. As you can see here, then the sales of Taxol decreased, while the sales of Abraxane significantly increased, and by 2020, it was estimated that the annual sales would be $2 billion. But what is Abraxane? It's nab-paclitaxel. Thereby, HSA is non-covalently bound to human serum albumin via hydrophobic interactions. The preparation is carried out via microfluidizer processing, and in the first step, HSA is bound to paclitaxel and primary aggregates are formed. These have particle sizes of 4 to 40 nanometers, and then form the nanoparticles, so nab-paclitaxel with around 130 nanometers. Within our study, we aim to investigate the applicability of Biotest HSA for the preparation of nab-paclitaxel. In addition, we wanted to compare the prepared nanoparticles with nanoparticles prepared with a commercially available recombinant HSA. Now coming to the workflow, we first of all performed design of experiment studies. For this, we used Biotest HSA and prepared blank nanoparticles. For the design of experiments, we chose the program model and created our DOE. As factors, we selected the amount of organic phase, which consisted of a mixture of ethanol and chloroform and was 0.6 to 6 milliliter. We added this organic phase to an aqueous phase with 1% of HSA and had a batch size of 30 milliliters. We then performed a pre-emulsification step at 8,000 to 12,000 RPM at a fixed time frame of 5 minutes. After that, we transferred the pre-emulsion to the microfluidizer. We here used the microfluidizer LM20 with a Z-type interaction chamber and performed 6 to 12 cycles at the homogenization pressure of 600 to 1,200 bar. After removal of the organic solvent, we characterized the particles regarding particle size, area of the peaks, and polydispersity index using photon correlation spectroscopy with a light sizer from Anton Bahr. After optimizing our process as well as our formulation, we prepared nab-paclitaxel. For this, we dissolved paclitaxel in the organic solvent, added it to the HSA solution, performed a pre-emulsification step for 5 minutes at 12,000 RPM, and then transferred this to the microfluidizer. There we performed 6 cycles at 1,200 bar and compared Biotest HSA with a commercially available recombinant HSA. After preparation, particle characterization was carried out regarding particle size and PDI via PCS. Zeta potential was investigated via laser Doppler electrophoresis coupled with PCS. Encapsulation efficiency and loading capacity was studied after separation of the water phase from the particles, and quantification was carried out via HPLC. We then also investigated shape and size of our nanoparticles. For this, we prepared blank nanoparticles and studied atomic force microscopy. Now coming to the results of our studies, for the blank nanoparticles, we saw that the particles were quite polydispersed and exhibited a quite broad particle size distribution. Thereby, we saw that we had a major size fraction of 120 to 300 nanometers, which can be assigned to the nanoparticles formed. However, we also had size fractions of monomer, dimer, and agglomerates. And for the generation of the DOE, we then decided to take the particle size and peak area of our main size fraction and to also choose the PDI as a response. First of all, we generated the coefficient plots, as can be seen here in this image. Thereby, generally, for a coefficient plot, it was significant if the error bar did not exceed our bar, and also if the bar showed a big difference from epsilon is 0. Regarding the particle size, we saw that the cycle number, as well as the interaction between the amount of organic phase and cycle number, had a significant effect on our particle size. Regarding polydispersity index, we found that the combination of the speed of our pre-emulsification step and the pressure during microfluidization significantly affected our particle size distribution. Thereby, high speeds of the pre-emulsification steps in combination with a high pressure led to small particle sizes and a narrow particle size distribution. Regarding the peak area, we saw that the most significant influence was the speed of the pre-emulsification step, as well as the amount of organic solvent and also the number of cycles. We then generated response counterplots of our DOE, as can be seen on the left side. We first studied the interaction between the cycle number and the amount of organic phase on our particle size. We used here a fixed setup of ultra-thorax speed of 12,000 RPM and a pressure of 1,200 bar and studied the size. We saw that the low cycle number in combination with a low amount of organic solvent leads to a small particle size. On the right-hand side, the response contour plots can be seen for a fixed cycle number of 6 cycles and a pressure of 1,200 bar. Thereby, we saw that high ultra-thorax speed in combination with a low amount of organic solvent led to a high peak area and a low particle size. Now coming to the results of our nab-paclitaxel studies, after optimization, we saw that the particle sizes of both HSA sources were in the range of 130 to 140 nanometers. We had a narrow particle size distribution with PDI values below 0.2. These results were comparable with our marketed product Abraxane. However, we saw that the particle size of Biotest HSA differed significantly from the size of the recombinant HSA. Then we had a look at our single batches and saw that the variations in particle size between the single batches were insignificant. However, we saw that the PDI was significant independent of the applied HSA source. However, here must be mentioned that after preparation, the particles can be filtered, which would reduce these significant values. We then subsequently had a look on the surface charge as well as our paclitaxel encapsulation. Zeta potential studies thereby confirmed that we had a moderate colloidal stability with values of 10 to 20 millivolt. The encapsulation efficiency showed values of 98% for both HSA sources and a loading capacity of around 9%. This is in line with the marketed product and shows that both candidates are promising for the preparation. We then studied atomic force microscopy of blank nanoparticles. We saw here that similar particle sizes as well as morphology of both HSA sources were obtained. Thereby, recombinant HSA showed particle sizes between 50 to 250 nanometers with particle heights of 30 to 90 nanometers, whereas Biotest HSA was in the size range of 30 to 200 nanometers and had particle heights of 30 to 110 nanometers. Here it must be mentioned that we measured AFM in the dry state and PCS results were generated in a liquid state. In conclusion, we found that the optimal parameters for the production of HSA could be determined via DOE. Thereby, a low amount of organic solvent in combination with the high speed of the pre-emulsification step, as well as a low number of cycles and a high pressure led to a narrow particle size distribution and low particle sizes. Drug loading with paclitaxel showed that both Biotest HSA is comparable to recombinant HSA, and both sources showed sizes of 130 to 140 nanometers with a narrow particle size distribution, a moderate colloidal stability, and a high encapsulation efficiency. The products are comparable to the marketed product Abraxane, and hence Biotest HSA is promising for the production of nab-paclitaxel. With this, I would like to acknowledge the funding of our research center through the COMET program and would like to thank all our colleagues and collaborators who have been involved in this project. Special thanks hereby go to Liye Maeyama from Biotest, Christina Schneider from Microfluidics, Ramona Jeitler and Eva Roblek from University of Graz. Thanks for your attention.
Thank you, Leah and Carolyn, for your interesting and very informative presentations. And thank you to everyone that has joined us so far for this webinar. Some references here from Leah's presentation, but other than that, Like I just mentioned, thank you for attending our webinar. Please contact either of our presenters or myself directly. And as mentioned in the chat, there's also a point of contact to further inquire about any questions you may have. But at this time, it looks like we have quite a bit of time remaining where we can go through some of the questions that were asked throughout this presentation. We'll likely start with the top. So let's see what we have here. So first question that was asked today was, would the microfluidizers be able to be sterile filtered? And the simple answer is yes, absolutely. Microfluidizer processors are regularly able to achieve particle sizes, distributions within any of those required parameters to be able to sterile filter. Another question that we have is, could you explain a bit more why CYS34 part of albumin is important? I believe that question would likely pertain to Liye.
Cysteine 34. This is an amino acid on the albumin molecule, and it has 3 different stages. One is the reduced, one is the reversible oxidized stage, and the irreversible oxidized stage. And we can say that the albumin antioxidant properties is due to this cysteine 34. And we can see in patients with morbidities like diabetes, like liver cirrhosis, the amount of oxidized albumin is much higher than the reduced form of albumin. So this meaning that the cysteine 34 is very important as scavenging free radicals, and usually in morbidities you have higher oxidation processes in the body. And the cysteine 34, when I talk to clients, I have not collected data yet, but it seems to play also a role in the nanoencapsulation processes. So cysteine 34, the state, the redox state, and also the dimer content. So in short, cysteine 34 shows this, it's a kind of parameter for the stability of albumin. The longer albumin is stored, the more oxidized it gets.
Thank you. Another question for you, Liye, would be, is 250 milliliters the largest configuration available? And do you have any plans to offer in liter or larger quantities?
Yeah, this is a question which I get often, because if you have a major production, of course, our clients would prefer bigger size. But I have to say, currently, we do not have any plans to offer bigger sizes. I think it will depend on the demand, but currently, unfortunately, there is no plan to offer bigger sizes.
Thank you. We have a couple other questions for you. One of them is, why is it a good point to have a lower fatty acids content for stem cells?
Yeah, this is a good point. You know, this area is not so much investigated, but certainly we can say that some of the fatty acids has a major role in induced pluripotent stem cells, because they tend to be very sensitive to low concentrations of molecules. And how much they play a role, specific fatty acids in the induced pluripotent stem cells, is not being investigated so much, but we can say that unfortunately a stabilizer we use in the albumin, which is a fatty acid, yeah, could have a negative effect on stem cells. So I think for fatty acid and the influence on stem cells needs more research, and we are starting to do this research with the effect of different kind of HSA, also including recombinant in induced pluripotent stem cells. This is a project we are running now.
Thank you for that. Another question, we'll likely ask a few questions for Carolyn now, just kind of flip the script a little bit. So Carolyn, one question that we had for you was, excuse me, I lost my train of thought. What is the percentage of Biotest HSA used or needed as excipients for a CAR T therapy? For example, in Kymriah, what would be the estimated percentage?
I think this is more related to Liye.
Oh, sorry,
For me.
apologies. Apologies, Liye.
CAR T cell therapy. To be honest, I need to check because in CAR T we have only 2 customers, and we are more in the induced pluripotent stem cell media. And for CAR T concentration, I would need to check. Sorry, I will get back to you.
Perfect. So Carolyn, now moving on to you a little bit. For your process, do you remove the solvent prior to high pressure processing?
No, we removed it after the microfluidizer processing. So after that, we removed it via steering, just normal steering in the hood. Since we had to perform everything in the highly potent room, we could not use a rotary evaporation, which is normally used.
And just to add on to that, in other applications for microfluidizers, processing with solvents in general, that is typically the case where it is evaporated or stirred out of these product afterwards, after processing, excuse me. Yeah. So another question for you that we have is, how does paclitaxel release from albumin?
So is this question related to release in vivo than in the human, or generally dissolution?
They did not seem to clarify.
So maybe then we in vivo, normally the particles are injected and then they separate again into these aggregates, and then they're usually taken up by the tumor. But to be honest, I have to clarify how exactly this works. So I think we could maybe come back to this afterwards.
Okay. On the same line of question, does albumin concentration affect the final size of the nanoparticle?
Yes.
Okay. And then what will be the concentration of chloroform or organic solvent in the final product? It seems like we have a few questions around the solvent removal process in general. So if you could just kind of touch on that for a few people in the chat, that would be great.
So general, we did not investigate the amount which is still present, but I think for sure for the marketed product there is some regulations how much chloroform ethanol is allowed to be there inside, but it should be removed afterwards. And then you also have lyophilization step afterwards. So prior to administration, the particles after preparation are lyophilized, and then they are resuspended in, I think, a buffer system before administration.
Thank you. We have a couple more questions before we wrap up. Along the same line of filtration, we have a question that says, did you try to sterilize through 0.22 filtration? What kind of membrane material did you use, and what was your filtration yield?
We did not filter it after preparation. We just used these special Amicon filters to filtrate or to separate our supernatant, the aqueous phase, from our particles to investigate the encapsulation efficiency.
Thank you. And then lastly, we have one more question that reads, which was the efficiency of conversion of HSA to HSA NP? Did you measure the residual monomeric polymeric HSA?
The encapsulation efficiency was 98% with a loading capacity of 9%. And regarding the second one, we measured the particle size, and there we saw that we only had one size fraction. In comparison to the blank nanoparticles, where we also saw size fractions of monomer and dimer, we did not saw it here in the paclitaxel formulation.
Thank you. Now, some of these questions may pertain to either Carolyn or Liye. So whichever one of you feels has the best answer, don't hesitate. So a couple more quick questions that are coming in last minute. We have someone would like to know the albumin stability with respect to pH. If so, what is the suitable pH adjusting agent?
The pH, you mean in the final product or in the nab-paclitaxel?
They don't seem to clarify which of those two. I guess whichever you believe makes more sense for the purpose of the question, or we can just move on.
Maybe we can get back to this question.
Sure. Yeah, that's fine. So another question that we have would be how fast monomeric HSA is formed after dilution?
Monomeric HSA after dilution, but this is also concerning the nab-paclitaxel formulation, or I don't know what, because in the product Biotest HSA, you have the liquid albumin, so more than 96% is albumin, and then you have, of course, the polymer and monomer content, yeah, and according to European Pharmacopoeia, there is a minimum of 90% monomer required. In the past, it was 95%, and then European Pharmacopoeia changed to 90%. But in fact, our specification says always we have a monomer content of more than 95%. I don't know if this answers the question.
I believe so for now. If not, we can always get back to them. But yeah, for now, we'll continue moving on. We have a couple more. So one question that we have is, which is the best method to remove chloroform, either TFF or rota evaporation?
I would say both are quite suitable. Usually in literature, they use rotary evaporation, but I think also TFF can be used.
Okay. Is Biotest excipients suitable for injectables?
No, Biotest HSA, Biotest HSA has GMP grade. It's pharmaceutical grade. We have the same manufacturing method like the pharmaceutical albumin. But because it's differently packed, it's not, it is an excipient packaging. Meaning, because the drug license, the part of the drug license is also the packaging, and because the packaging is not pharmaceutical, how you say, it's not a drug license packaging, it cannot be infused to patients. It must not be. It is only for manufacturing use. But
thank you for that clarification.
Yeah, I just want to emphasize that all the documentations, because it's the same process and the same line, it's only differently labeled, let's say like this. All the documents are the same as the European Pharmacopoeia requirements. So we have all the same documentation as for the pharmaceutical.
Okay, thank you for that clarification. And is Biotest HSA a BLA application in US FDA? Does that make sense?
Yeah, it makes sense. Biotest HSA has EP, so it's based on European Pharmacopoeia, and we do not have the drug license in the US. We are considering a drug master file category type 2, which means the module 3 in the dossier. And we made a gap analysis internally, and we can say, because Biotest HSA is actually a product which is EMA approved. So it is approved by the European authorities. We can be confident that this drug master file type 2 would fulfill the FDA requirements. But what I can say now is it's not a drug licensed product in US.
Thank you. A couple more quick questions. We only have 5 minutes left, so please try to get your questions in if you have any that you're hesitating to ask. Otherwise, as a reminder, the webinar is recorded. All questions that weren't answered here can be answered later, or feel free to follow up via the contacts on our screen or with our channel partners or website or any of those avenues. So just a couple more, as I mentioned, what is the optimal initial aqueous phase HSA concentration for 130 nanometer HSA PTX nanoparticles?
We used 1% HSA, which is also in line with the patent for the nab-paclitaxel product, and we used a batch size of 30 milliliter.
Thank you. And do pre-saturization with solvent required? Is pre-saturization with solvent required for Biotest HSA? And if yes, why?
Yes, we pre-saturated it. This was according to our literature review. We found that pre-saturation is more efficient if you pre-saturate it before, and we then decided to use only pre-saturated aqueous phase.
Thank you. And we have one more clarification question from earlier. Relating to the release question, could you explain the in vivo process? I know we were wondering what they were asking about, but if you could explain the in vivo process a little bit and also how the encapsulation efficiency was measured.
Regarding in vivo, as far as I know, it's active transport inside the cell, but maybe to have a clearer view, we can answer this question later on to have really according to. And regarding encapsulation efficiency, we used special filters, centrifuged our suspension, then used the aqueous phase for quantification via HPLC, and then determined the encapsulation efficiency.
Okay, so we have a few more last minute biotest questions. So are HSA NPs stored in solution or as a solid lyophilized material?
Biotest HSA is a liquid. Yeah, yeah.
And can Biotest HSA be used as an excipient? I think that was breaking off of an earlier question.
It can be used as an excipient, but regulatory-wise, of course, you need to be aware that if you use it as an excipient in your final product and you submit, you go for IND or BLA, then you would need to have all the data which is required in the module 3, I think, in your dossier for IND and BLA. In this module 3, I think you would need to integrate many informations on the excipients, including Biotest HSA. And but we are selling actually Biotest HSA in the US as an excipient, in the excipient packaging. But regulatory-wise, you would need to know all the, let's say, the procedure until the submission of IND and BLA or in a new drug application. Yeah.
Thank you. So at this point, we're at the top of the hour, so I'm going to hold off asking any more questions to our speakers as we wrap this up. I just wanted to say another big thank you to our speakers, Liye and Carolyn, for joining us and for presenting all the information that you have today. And huge thank you again to everyone who has joined us online and to those that will view this webinar later. The link for the webinar will be shared with everyone shortly, and there should be Plenty of avenues to be able to obtain a copy of this presentation as well for those of you that asked in this chat. But other than that, we hope everyone has a great day. Thank you for joining us, and so long. Thank you very much.
Thank you. Matt.
Thank you.
Thanks. Bye.